In this video, we'll be covering the urea cycle for the purposes of Step 1, as well as in-house exams. On exam day, the urea cycle is one of those high-yield biochemistry topics that you're almost guaranteed to encounter, whether it's enzyme defects, nitrogen metabolism, or ammonia toxicity. Understanding this pathway is absolutely crucial. To start with, what is urea? Urea is basically a waste product of protein metabolism. Specifically, when protein is consumed, it is broken down into amino acids which contain nitrogen. Now, when these amino acids get metabolized, the nitrogen that's contained within them gets released in the form of ammonia, a highly toxic substance to the human body, if it happens to build up. The body's way of preventing this buildup is through the detoxification of ammonia in the form of urea, through the urea cycle. Urea is synthesized in the hepatocytes of the liver, involving the mitochondria and the cytoplasm, after which it is excreted in the urine by the kidneys. Now, let's suppose that there's a problem in the urea cycle, which is preventing the formation of urea. Because of this problem, ammonia can no longer be detoxified through the urea cycle, and instead, it will build up in the blood to toxic levels, leading to a condition known as hyperammonemia. To pinpoint where the issue is in the body, we can use something called the BUN, or blood urea nitrogen, which measures the urea levels in the blood. A low BUN means that urea production is reduced. This suggests liver failure, because as we've already stated, the liver is the organ that is responsible for converting ammonia into urea. And if we can't do that, then we'll have a buildup of ammonia. On the other hand, a high BUN means that urea levels are increased. This means that the liver is working perfectly fine to produce urea. However, the kidneys are failing to excrete it, causing urea to build up in the blood, a condition known as uremia. Both hyperammonemia and uremia are conditions that involve disruptions to nitrogen metabolism, and they can have overlapping features. We'll look at them in a bit more detail right after we cover the urea cycle pathway itself. The urea cycle starts off in the mitochondria of hepatocytes, where ammonia and bicarbonate serve as the starting substrates, which combine to form carbamoyl phosphate. This reaction is catalyzed by the enzyme carbamoyl phosphate synthetase 1, abbreviated as CPS-1. CPS-1 is particularly high yield because it is actually the rate-limiting enzyme of the urea cycle. The USMLE loves asking about rate-limiting enzymes of different biochemical pathways, so make sure to have this memorized. Another high-yield point to remember is that CPS-1 requires an obligate activator called N-acetylglutamate in order for CPS-1 to function. This means that without NAG, CPS-1 would be unable to convert ammonia into carbamoyl phosphate. Next, carbamoyl phosphate is converted to citrulline. I'll discuss the enzyme responsible for this step later in the video, but for now, just remember that this step forms citrulline. Remember, at this stage, we are still in the mitochondria, but once citrulline is formed, it crosses the mitochondrial membrane and enters into the cytoplasm, where it is further metabolized into arginosuccinate. This reaction is catalyzed by the enzyme arginosuccinate synthase. This is an energy-dependent process driven by the hydrolysis of high-energy phosphate bonds in ATP, which converts ATP into AMP. Now, here's the high-yield point that's often overlooked in other sources. This is the step where the second nitrogen atom of urea is added. This nitrogen comes from the amino acid aspartate, joining the first nitrogen, which was donated by ammonia in the mitochondria. This is how urea, the final product of this cycle, contains two nitrogen atoms, one from ammonia and one from aspartate. Next, arginosuccinate gets converted to arginine by the enzyme arginosuccinase, otherwise known as arginosuccinate lyase. This reaction also produces fumarate as a byproduct. An additional point which you could peripherally know is that fumarate then goes on to participate in the TCA cycle, aka the Krebs cycle, as an intermediate. Now, for the key step that gives the urea cycle its name, the conversion of arginine into ornithine, which produces urea as a byproduct. Urea then goes on to be excreted by the kidneys. The enzyme that's responsible for this step is arginase. As the name suggests, this enzyme breaks down arginine. Next, ornithine crosses the mitochondrial membrane to enter the mitochondria, where it combines with carbamoyl phosphate to form citrulline. This enzyme that catalyzes this reaction, which we previously skipped over, is called ornithine transcarbamylase, abbreviated as OTC. And with that, we've now come full circle. This, in essence, is the urea cycle. I know it's a lot of long and complicated names to remember, and you're probably wondering that there has to be an easier way to recall all of this on exam day when you're going to need it the most. And don't worry, because I have the perfect mnemonic that will help you not only remember the intermediates of the cycle, but also their specific locations within the hepatocyte itself. The mnemonic I've created for this is, occasionally, silly arguments make urea. And here's how it works. The word occasionally represent the steps that occur in the mitochondria, while the words after it, silly arguments make urea, represent the steps that take place in the cytoplasm. To make it even easier to visualize, I've color-coded the locations. Purple for the mitochondrial steps and pink for the cytoplasmic steps. This way, you can easily remember both the sequence of events as well as where they happen. The mnemonic starts off with occasionally, representing the mitochondrial steps, where O for ornithine and C for carbamoyl phosphate combine to form C for citrulline. It is important to note that I didn't include ammonia and bicarb in the mnemonic, as it's already clear that this is the starting point of the cycle. Of course, given that the main focus of the urea cycle is to detoxify ammonia. Next, silly arguments make urea correspond to the cytoplasmic steps. I chose to represent the S in arginosuccinate with silly, since A for arguments naturally represents the next step, arginine. Finally, of course, urea in the mnemonic corresponds to the last step where urea is formed. If you take some time out to memorize this mnemonic, even the enzyme names will just come naturally to you. For instance, arginosuccinate synthase will of course have arginosuccinate as the product of that reaction. So if we just know the intermediates to this mnemonic, we basically already know the enzyme names as well. This mnemonic, along with the second half of this video, helped me in immensely improving my understanding of the urea cycle and how it presents on step 1. Now that we've covered the urea cycle, let's take a closer look at its rate-limiting enzyme, CPS-1. As the name suggests, CPS-1's role is to synthesize carbamoyl phosphate. But what about the 1 in CPS-1? This distinction is important because, as you might expect, if there's a CPS-1, then there must also be a CPS-2. And this slide will break down the key differences between these two enzymes, which again is a high-yield topic for exam day. To begin with, both of these enzymes act as the rate-limiting enzymes for their respective cycles, CPS-1 for the urea cycle and CPS-2 for pyrimidine synthesis. I'll cover pyrimidine synthesis in a separate video, but it's important to note this distinction now. When it comes to location, CPS-1 is found in the mitochondria, as we already know from our urea cycle mnemonic, which starts off with occasionally, which corresponds with the mitochondrial steps. CPS-2, on the other hand, is located in the cytoplasm. To remember this, just think of cytoplasm instead of cytoplasm. The last difference between these two enzymes is the fact that CPS-1 requires NAG as an obligate activator, whereas CPS-2 doesn't require any activator. While these two enzymes have considerable differences, they share one very high yield similarity that we actually already know. They both synthesize carbamoyl phosphate, as their name suggests, of course. This shared product, carbamoyl phosphate, is absolutely vital to understanding how the urea cycle and pyrimidine synthesis pathways actually connect and interact with one another before they go their separate ways. To illustrate what I just said, Let's imagine that we knock out an enzyme in the urea cycle after carbamoyl phosphate has been synthesized. This will cause carbamoyl phosphate to build up, since it can no longer go through the defective urea cycle. Instead, the excess carbamoyl phosphate will spill into the cytoplasm and be diverted to an alternative functional pathway, pyrimidine synthesis. In doing so, there's overproduction of one particular toxic intermediate, orotic acid. This leads to a condition known as secondary orotic aciduria. The term secondary indicates that the rise of orotic acid in the urine is secondary to the urea cycle being defective. This crossover from the urea cycle to the pyrimidine synthesis pathway, and I'm emphasizing this again, happens only after carbonyl phosphate is synthesized, as it is the only intermediate that is shared between these two pathways. This is very high yield to understand. Let's look at a high yield example of this to understand what I just said. So let's imagine that we knock out an enzyme like OTC. If we knock out OTC, carbamoyl phosphate can no longer proceed through the urea cycle. Thus, both carbamoyl phosphate and ammonia will build up, since they are upstream to the defective OTC. This, as we already know, leads to hyperammonemia. On the other hand, all intermediates that are downstream of the defective OTC, including urea, will decrease. At the same time, when carbamoyl phosphate builds up in the mitochondria, because it can no longer enter the urea cycle, the excess spills into the cytoplasm. As I've discussed before, this carbamoyl phosphate is then diverted into the pyrimidine synthesis pathway, leading to the overproduction of orotic acid, resulting in secondary orotic aciduria. To summarize, if we knock out OTC or any other enzyme that's downstream of carbamoyl phosphate, we'll see two major effects: hyperammonemia and orotic aciduria. And remember, this is only made possible because carbamoyl phosphate is a crossover point between the two cycles. Now that we've understood what happens when an enzyme that's downstream of carbamoyl phosphate is knocked out, let's consider a different scenario. What if the enzyme that's responsible for synthesizing carbamoyl phosphate itself is defective? This enzyme, CPS-1, catalyzes the first step of the urea cycle. If CPS-1 is defective, carbamoyl phosphate can no longer be formed. Additionally, all other downstream intermediates of the urea cycle will also be absent. On the other hand, intermediates that are upstream to CPS-1, particularly ammonia, will build up, leading to hyperammonemia. And unlike a defect in OTC or an enzyme that's downstream to carbamoyl phosphate, there will be no orotic aciduria, since carbamoyl phosphate is not even produced in the first place, and therefore a crossover to pyrimidine synthesis cannot occur at all. All that we've been discussing so far, the differences in what happens if CPS-1 is deficient versus if OTC is deficient, is incredibly high yield. To summarize the differences between them, I've defined these differences in a table format, and here are the differences that I've already discussed. One high yield that I haven't discussed yet is the inheritance pattern of these enzyme deficiencies. In genetics, I want you to know that it's a general rule that most enzyme deficiencies follow an autosomal recessive inheritance pattern, and this is true for CPS-1 deficiency. However, OTC deficiency is an exception to this rule. It follows an X-linked recessive inheritance pattern. Because this is an exception to the general rule, this makes OTC's inheritance pattern an extremely high-yield detail to remember. Lastly, for the symptoms, as we've already discussed, both will lead to hyperammonemia. However, because OTC deficiency also leads to orotic aciduria, it is associated with urinary symptoms as well, such as dysuria and precipitation of orotic acid crystals in the urine, which may appear as cloudy urine or sediment. Now, throughout this video, we've mentioned hyperammonemia as a key feature of urea cycle disorders. But we haven't really explored why ammonia accumulation is so harmful. Before diving into its clinical presentation, let's first look at the pathophysiology. One way that ammonia harms the body is by interfering with energy production. While the urea cycle is the main pathway for ammonia detoxification, there's one more mechanism which you should be familiar with, and that's glutamate synthesis. As you can see, this process not only uses ammonia, of course, but also uses alpha-ketoglutarate, which is an intermediate of the TCA cycle. Normally, this is not an issue. However, if the body is dealing with excess ammonia, then that would mean that more alpha-ketoglutarate would be needed towards glutamate synthesis to detoxify ammonia. This depletion of alpha-ketoglutarate from the TCA cycle, which is the main pathway for producing ATP, the body's energy source, leads to energy deficiency symptoms that we'll discuss shortly. Additionally, detoxifying ammonia via glutamate synthesis alters the body's glutamate levels. And since GABA is derived from glutamate, we will also have altering GABA levels. This will affect both excitatory and inhibitory signaling, respectively, in the brain. Now, I'm sure you can see how depleting alpha-ketoglutarate triggers a whole cascade of biochemical disturbances, leading to a wide range of symptoms. Along with altered levels of glutamate and GABA, hyperammonemia also causes changes in glutamine levels, since glutamine is derived from glutamate. The problem with glutamine accumulation is that it acts as an osmotic agent, which means that when glutamine builds up in the brain, it attracts water, leading to cerebral edema. To easily remember all of these biochemical disturbances, just focus on the letter G, starting with alpha-ketoglutarate to glutamate, which then gives rise to GABA and glutamine. Altered levels of these key quote-unquote G molecules leads to the hallmark neurological symptoms that we see in hyperammonemia, including confusion, slurred speech, blurred vision, lethargy, seizures, and in severe cases, coma. The most specific neurological finding for hyperammonemia is Asterixis, also known as the flapping hand tremor. Asterixis can be observed when the arms are extended and the wrists are bent back or dorsiflexed, which causes sudden, jerky downward movements of the hands that look like a bird flapping its wings, as seen in the image above. Asterixis is the most specific finding for hyperammonemia, and if you see anything like this in a question stem, you should automatically think of hyperammonemia. Now that we've covered the clinical presentation of hyperammonemia, let's move on to treatment. Since ammonia is derived from protein, if we reduce protein intake in the patient, we will automatically observe a decrease in both ammonia and urea levels. Alongside dietary modification, we can also use two high-yield medications, lactulose and rifaximin. Lactulose works by being fermented by gut bacteria into acidic metabolites. These acids convert ammonia, NH3, the absorbable form, into ammonium, NH4+, the non-absorbable form. This ultimately reduces ammonia absorption. Rifaximin, on the other hand, is an antibiotic that lowers ammonia levels by directly targeting and killing ammonia-producing bacteria in the gut, and thus it lowers both ammonia production as well as absorption. Both of these drugs have simple mechanisms of action, but are very high yield, especially lactulose. And that wraps up our discussion on the urea cycle. Thank you so much for sticking around until the end of the video. It means a lot. This is my first upload, and I'd really appreciate if you could like and subscribe, as I'll be sharing more high-yield content soon. I know I have a lot to learn when it comes to content creation, so if you have any feedback, feel free to drop it in the comments or send me an email. Thanks again, and I'll see you in the next one.